Okay, so welcome to this next video in which we are discussing type 2 activation of endothelial cells. Okay, so far what we've seen is that when these sentinel cells, such as macrophages and dendritic cells, sense the presence of an intruder, such as Staphylococcus aureus, via the detection of the pathogen-associated molecular patterns, then what they're going to do is release two pro-inflammatory cytokines, tumor necrosis factor alpha and interleukin 1. These two pro-inflammatory cytokines are going to trigger type 2 activation of the endothelial cells on the arterioles, the capillaries and the venules in the affected area. We're now at the moment looking at the pathway associated with TNF-alpha. So TNF-alpha binds to the tumor necrosis factor receptor 1, okay, and that causes a change in conformation of the tumor necrosis factor receptor 1, which then allows it to associate with another protein. So another protein is going to be recruited to the intracellular domain of the tumor necrosis factor receptor 1 once it has bound tumor necrosis factor. Okay, and this next protein is the tumor necrosis factor receptor associated via death domain protein, okay, or the TRAD protein. Once the TRAD has bound to the tumor necrosis factor receptor 1, then what will happen is the serine threonine kinase receptor interacting protein 1, RIP1, will then also be recruited. Finally, another protein, the tumor necrosis factor receptor associated factor 2, TRAF2, will associate with RIP1, and this will create something known as the signalosome. Okay, now the signalosome is going to activate two transcription factors. One of these is the activator protein 1. The other is going to be NF-kappa B, and we were just discussing how NF-kappa B is going to be activated. So, in a normal cell, NF-kappa B is kept inactive by having another protein, the inhibitor of kappa B, bound to it. Okay, so now let's show NF-kappa B. So NF-kappa B is actually a dimer of two separate proteins bound together. Okay, and these two separate proteins that are bound together to make NF-kappa B are a protein known as P50, okay, which will that be this one here, and another protein known as P65. So the dimer of P50 and P65 together, these two are together known as NF-kappa, so you put a little K there, B. Okay, so this means nuclear factor, so it's going to be a transcription factor, and it's nuclear factor kappa B. Okay, right, so this NF-kappa B, which consists of this P50 with this P65, usually is kept inhibited by having I-kappa B, the inhibitor of kappa B, bound to it. Now, in order to activate the nuclear factor kappa B, what you have to do is get rid of this inhibitor of kappa B, basically. It has to be removed from the NF-kappa B, and then the NF-kappa B, this dimer of P50 and P65, can translocate into the nucleus and act as a transcription factor. Okay, so basically this signalosome is going to activate a whole bunch of signaling cascades that leads to both the activation of activator protein 1 and the inactivation of inhibitor of kappa B, so that you get the activation of NF-kappa B, nuclear factor kappa B. Okay, now we'll discuss what the activator protein 1 and the nuclear factor kappa B, or this dimer of P50 and P65, are going to do within the nucleus later, because the interleukin 1, this other pro-inflammatory cytokine, is also going to lead to the activation of the activator protein 1 and the nuclear factor kappa B. So we might as well discuss the pathway by which interleukin 1 activates these as well, and then we can discuss activator protein 1 and nuclear factor kappa B together, okay? And it will cover both tumor necrosis factor alpha and interleukin 1. Okay, so let's now discuss how interleukin-1 acts on these epithelial cells, or these endothelial cells, to cause type 2 activation. So, if we have the basolateral membrane again of our endothelial cell here, okay, and we'll have another receptor here, and this is going to be a receptor for interleukin-1. Okay, and we've discussed that the interleukin-1 receptor that these endothelial cells will have is the type 1 interleukin-1 receptor. So on the basolateral membrane of our endothelial cell, we have the type 1 interleukin-1 receptor. Okay, and this is usually denoted as the IL-1 for interleukin-1. Okay, and then you put an R for receptor, and then you put another 1 to denote that this is the type 1 interleukin-1 receptor. Okay, so let's give the type 1 interleukin-1 receptor a colour. So we'll let it be blue here. Okay, and now what's going to happen is that the interleukin-1 from the resident macrophages or the dendritic cells is going to come and bind to this type 1 interleukin receptor. So here is the interleukin-1. So this is interleukin-1, and I think I will write interleukin-1 out in full. Okay, so this is interleukin-1. Okay, and I'll colour that in red. Okay, so what follows now? Well, what's going to happen is that interleukin-1 receptor, type 1, is going to now get a bunch of proteins that are going to come and associate with the cytoplasmic domain now. So once the ligand has bound to this type 1 interleukin-1 receptor, it's going to change the conformation of the intracellular domain, and that's going to allow other proteins from within the cytoplasm to come and associate with that intracellular domain. So one of these proteins that's going to come and associate, so I'll draw, there are going to be two. Two are going to come and bind to this type 1 interleukin-1 receptor, so I'll split these up here. Now one of them is what's known as the myeloid differentiation primary response gene 88. Okay, so this is the myeloid differentiation primary response gene 88. Okay, and this is often abbreviated to MYD88, so the myeloid differentiation primary response gene 88. Okay, right, and as I say, that's often abbreviated to MY for myeloid differentiation, and then usually this is done with a capital D here. So you have MYD and then 88. So MYD 88. So we'll colour in MYD 88 in this pink colour here. Okay, so this is one of the proteins that's going to come and associate with this type 1 interleukin-1 receptor, MYD88. Okay, the other protein that's going to come and associate, this one here, is the Toll/interleukin-1 receptor accessory protein. Okay, so the Toll/interleukin-1 and then receptor accessory protein. Okay, and again, because this is a mouthful of a name, it's often, oh dear, what have I done? I've drawn the name of this right through where I want to show the next proteins. That's annoying. Okay, never mind, I'll just have to try and rectify it. The Toll/interleukin-1 receptor accessory protein. Okay, and this is often abbreviated to the TIRAP. So you take T for Toll, you take the I from interleukin-1, you take the R from receptor, you take the A from accessory, and you take the P from protein, and you get TIRAP. So this protein here that's also come and associated with the cytoplasmic domain of the type 1 interleukin-1 receptor is known as TIRAP. Okay, so I will colour this in turquoise. Now, the TIRAP protein is also often called the MAL protein. Okay, so MAL is also another name for this TIRAP protein. Right, so we've had these two proteins, the myeloid differentiation primary response gene 88, the MYD88, and the Toll interleukin 1 receptor accessory protein, the TIRAP or MAL protein, come and associate with the cytoplasmic domain of our type 1 interleukin 1 receptor. We're now going to get two more proteins which are specifically going to come and associate with the myeloid differentiation primary response gene 88. So I'll try and squash these in here. Okay, so there are two proteins that are going to come and bind here. And thankfully they have very similar names. So the first one is going to be a protein known as interleukin 1 receptor associated kinase 1. Okay, so where should I put this? I'll try and put it here. So the interleukin 1 receptor, so I'll just put IL-1R, associated kinase 1. And again, this has an acronym. So you take the I for interleukin 1. Okay, so there's the I. You take the R from receptor, you take the A from associated, and let me just move this up a little bit. Okay, and you take the K from kinase to get Iraq, and then 1. Okay, IRAC1. Okay, so IRAC1 is going to be one of these proteins that's going to come and associate here. And I think I'll cover IRAC1 in orange. So here is the protein IRAC1, the interleukin 1 receptor associated kinase 1. Okay, and then the next protein that's going to come and associate is the IRAC4, so the interleukin receptor associated kinase 4. Okay, and we'll show that in blue here. So, where do I want to put this? So, I will try and label these up in the best way I can. So this is the interleukin 1 receptor associated kinase 4 in here. And the orange one is this interleukin 1 receptor associated kinase 1. Okay. Now finally, one more protein is going to come and associate with the interleukin 1 receptor associated kinase 1, and I'll try and squeeze this in here. So one more is going to come and bind there, and this final protein is what's known as TNF receptor associated factor 6. Okay, so TRAF6. So just like we saw up here, we had TRAF2, which was TNF receptor associated factor 2. Down here, we're going to get, let me put this down somewhere, TNF receptor associated factor 6. Okay, and for short, this is abbreviated to TRAF6. Okay, so TRAF6 is going to come and bind to the interleukin 1 receptor associated kinase 1, IRAC1. Okay, so this is TRAF6. Right, so now we have assembled another great big signaling complex, which is also going to activate the exact same things as the signalosome did in the case of TNF-alpha. So now what's going to happen is that TRAF6, well in fact the entire signaling complex, but specifically this complex consisting of the myeloid differentiation primary response gene 88, the interleukin 1 receptor associated kinase 1, the interleukin 1 receptor associated kinase 6, and also the TNF receptor associated factor 6, all of those, that complex there, is going to now activate. So let me show this activation. It's going to activate the activator protein 1, AP1, and it's also going to inactivate the inhibitor of kappa B, I-kappa B, which will then lead to the activation of nuclear factor kappa B, which remember is this dimer of P50 with P65. Okay, so the overall result of both of interleukin 1 and tumor necrosis factor alpha, then is to lead to the activation of the activator protein 1 and also of this nuclear factor kappa B, and both of these are transcription factors. So what they're going to do is alter the gene expression within this endothelial cell, and that altered gene expression is everything that type 2 activation of endothelial cells is. It's the beating soul of type 2 activation of endothelial cells. Okay, now we'll see what the nuclear factor kappa B and the activator protein 1 are going to do in the next video.